excessive talk after Cliff.
Greetings, everyone. My name is Katie Edwards. I'm from the University School of Pharmacy and Pharmaceutical Sciences. I'd like to present our work on liver metabolism in both brook trout and lake trout in collaboration with my colleagues at Cornell. The only thing I have to differ on with Cliff, since I come from the School of Pharmacy, is vitamin B1 deficiency is not necessarily obsolete in our population, so take your vitamin B1. We're really focusing on liver metabolism and how it's organized differently between different species and strains, and how it changes upon exposures to environmental stresses. So we're focusing on things such as carbon efficiency, redox defense, and ability to have a reserve capacity under different conditions. So we essentially developed a panel of assays, both ranging from morphometric and biochemical assays along the oxidative and non-oxidative pentose phosphate pathways, as well as redox metabolism for glutathione and its recycling capacity. Glutathione is an important antioxidant. It's an endogenous molecule that's made of amino acids. So ultimately, we were initially looking at how these biochemical parameters relate to morphometric parameters. This was carried out on hatchery-reared brook trout under ambient elevated temperatures and acute hypoxia conditions. Also some hatchery-reared lake trout and some lake trout from Lake Ontario in the Sudbury region of Canada that were leftover livers in my freezers from Brian Neff's group. And essentially, the very first thing in order to carry out these assays is we first need to extract the protein. Enzymes are proteins, so we need to get that out. So we weren't really thinking that that was necessarily all that interesting, but it turned out some unique relationships showed up. So first off, basically, you're probably familiar with the hepatosomatic index, which is the ratio of the mass of the liver to the mass of the body mass. So a larger hepatosomatic index would indicate that the fish is allocating more resources to developing the liver. But on top of that, what is the liver composed of? So just as part of our natural workflow, we needed to extract the protein, determine the protein concentrations, and ultimately we found that under identical rearing conditions for different strains of brook trout, we saw very different protein densities in the livers. So for example, the LT strain had a very dense protein liver relative to, say, the ASN strain. So right off the bat, that tells us there are some strategic allocation differences in between livers, between fish. And the liver is a really important organ for redox metabolism and metabolism in general for nutrients. So when we have a lower protein concentration, that indicates that that mass of the liver is allocated to other things. Maybe glycogen, maybe water, what have you. So hepatosomatic index alone does not predict the function of the liver. This is a change in architecture of the liver. It's not necessarily related to scale. So biochemically, we took this protein extracts and we're now looking for enzyme activities. One of our primary foci was the pentose phosphate pathway, which does a number of things, but you can think of it as a shunt for sugar metabolism under anaerobic metabolism, among other things. Two main phases. One is the oxidative phase, which is controlled by glucose 6-phosphate dehydrogenase. Secondly is the non-oxidative phase, is controlled by transketolase, which is a thiamine-dependent enzyme. The relative amounts of these processes depend on the needs of the tissue. Why do we care about these pathways? Well, first, the oxidative phase produces what's known as R5P. R5P is needed for RNA and DNA synthesis, which is needed for successful growth, reproduction. Another key product of the oxidative phase is NADPH, which is a critical reducing equivalent in cells, also for the production of cholesterol synthesis, fatty acid synthesis, and metabolism. So the type of assays we're carrying out are primarily kinetic-based enzyme assays. So we take that solution phase extract of the protein and we're adding whichever reagents we need to measure a given enzyme. And this is some example data. I wish this was bigger. So essentially what we're doing, this is a transketolase assay. We're measuring the activity of the enzyme in the presence of the fish's endogenous levels of thiamine. And then we're seeing how that rate changes if we supplement the tissue extract with thiamine. So the rate in blue indicates the endogenous activity. The rate in red indicates the capacity. So that basal transketolase activity is reflective of the expression level, the stability, and the availability of cofactors, whereas the saturated transketolase rate is really the Vmax. What is the capacity for that tissue to produce what it needs to by way of the non-oxidative pathway? So this activity is thiamine dependent. This is different thiamine concentrations, and I'm going to point here with the fish. Different thiamine concentrations as a function of rate. And so we can see the concentration dependence. This is 2 different strains of lake trout, and we can see different maximal activities. We can also get affinity measurements. How much on the protein-ligand interaction, how much time is needed to saturate that, relatively speaking, strain to strain, species to species, what have you. The latency is another descriptor, which is basically what's the excess capacity that we're not using. So some interesting things popped out of this. We carried this out over a variety of different lake trout and brook trout, both hatchery and native environments. And recalling that higher capacity is essentially more unused capacity for the non-oxidative pentose phosphate pathway. And there are essentially 3 regimes that we observed. One of which is that we have, say, high basal activities over here with high latencies, so high on the y-axis. So that indicates that the fish it's got sufficient thiamine, whatever that means for a given species, and capacity to respond to additional dietary amounts. On the flip side, though, when we scale over here into looking at low basal activities, this part of the axis, with low latency, that indicates that it's usually suggestive of a prolonged thiamine deficiency, and no matter how much thiamine you give, that fish is not going to be able to respond. So measuring thiamine levels alone tells you dietary intake, but it doesn't necessarily tell you how the fish is going to be able to respond to that. This is particularly important when we start to look at not necessarily eggs, because these pathways are not developed at this point, but because we're looking at adult liver tissue. So that's the transketolase being the non-oxidative phase. We're interested in the oxidative phase because that is really the primary mechanism for most tissues to produce NADPH. One thing to take away from this is that the balance between oxidative and non-oxidative metabolism may also be due to a nutrient limitation. So you can go both pathways and still end up making R5P for RNA and DNA. But we're losing one molecule of carbon by way of CO2 through the oxidative phase. So under nutrient-limited conditions, a fish may not physiologically want to pursue the oxidative phase. The non-oxidative phase bypasses that. So it allows us to essentially recycle rather than have carbon loss. So that may be an important consideration as we move forward and think about the balance. So thiamine's importance as a transketolase factor may under overall low nutrient conditions may be elevated. There may be more dependence on the transketolase activity in non-oxidative pathway. So ultimately, we looked at this across a variety of, in this case, 5 different brook trout strains, seeing that most of them did not show much difference in their basal transketolase activity under endogenous conditions. TEM being an exception, it did have significantly lower activity. And then when we supplement those fish tissues with high degrees of thiamine diphosphate, we can see what's the maximal capacity. And we can see that in the environment, like what happens if they have high food availability and they take in something besides alewives. Are they able to respond? So that's where that matters. So we have then also pursued the oxidative phase of this assay, looking at G6PDH. And the strain differences were one thing in brook trout. But what was really remarkable is that when we looked at brook trout versus lake trout, the activity of both phases of the pentose phosphate pathway, both the oxidative and non-oxidative, were much greater for brook trout than for lake trout. So the brook trout are more likely needing to adapt to more metabolic agility, maybe low oxygen conditions and what have you, than do lake trout, maybe perhaps a little more stability. And I'm really thinking about this mainly from a biochemical standpoint. So huge differences in metabolism in brook trout and lake trout. The other piece we mainly looked at is redox strategies, where we're looking at, again, at reduced glutathione, and that's critical for mediating reactive oxygen species, mediating the phase 2 metabolism of xenobiotics. And in order for that glutathione to be in the reduced phase where it's active, we need glutathione reductase. So it's an enzyme that cycles between the oxidative and reduced phases. Our data suggests there are two distinct strategies for redox metabolism. One, we either have a high recycling capacity and essentially a low pool of glutathione. The other is that we are generating a huge pool of glutathione and yielding high buffering capacity. So the latter has an energetic cost. Those amino acids that are used to make glutathione, they can be used for something else. So that's not necessarily the fish's best course if they aren't exposed to a lot of reactive oxygen species. Between the brook trout, we saw that there were differences between strains in the recycling capacity, whereas lake trout, we only looked at Seneca and slate strains. There wasn't much difference there. But then when we moved from a hatchery to a wild environment, we saw that lake trout have a significantly higher GR capacity than brook trout. Both raised in the hatchery environment. The conditions aren't one-to-one, so I can't speak too much to that. But really what was notable is taking lake trout from a hatchery environment and putting them into a different fish, but collecting them from the same strains from a Lake Ontario versus hatchery. There was a lot higher glutathione reductase activity in the wild. So they are really ramping up their ability to recycle that glutathione. It's a great strategy to minimize their expense in terms of glutathione pool. Where that made a big difference is also in the glutathione pool. Strain differences between brook trout existed in terms of the amount of that glutathione pool. Some were more or less willing to dedicate resources to it. But we couldn't even detect glutathione in lake trout. The levels were so low. So lake trout put nearly all of their emphasis into that enzymatic recycling. At the end of the day, so we've carried this out in full for our brook trout. The lake trout are just something we're just starting with. There are differences in strain for each parameter. So this plot shows the different parameters we've measured. The strains are on the y-axis. The darker red means that under the relative conditions, it has the most of that activity or the most of that compound. Blue meaning the least. Just to point out a couple of these, we can see that the TEM strain for brook trout has a limited capacity for the non-oxidative phase of the pentose phosphate pathway, and that's highlighted in the teal. You see a lot more blue. But it has pretty strong oxidative pentose phosphate capacity. These fish were raised under identical rearing conditions, same light, same diet, same temperature, versus TXD, which had a moderate non-oxidative pentose phosphate pathway, realized flux, and really high capacity for expansion. Thinking about the glutathione axis, we see TEM has a limited capacity for both the pool and recycling, whereas TXD had a large glutathione pool and a high recycling capacity, which would suggest that it has a good resistance to oxidative stress. So this overall likely guides resilience and long-term adaptation to their environments. We then, to simplify all these points, we compress this data into three axes. One of which is the ratio of the oxidative pentose phosphate capacity to the realized oxidative pentose phosphate capacity. Another is the maximal capacity of transketolase versus its basal. And the third is that glutathione pool versus the recycling. Main thing I want to point out is these strains really differ quite a bit in the space that they occupy in this metabolic matrix. So strains have markedly different strategies. When we move this on to lake trout, again, all the brook trout tend to be on the right-hand side of this right-hand side of the graph, where our lake trout tend to be on the, here's all of our lake trout in this part. So, significantly different metabolic strategies between species and between strains. So we're also starting to see some locational differences in the lake trout, so that's something we're just starting to have a look at. So we're really seeing that metabolic architecture is going to reflect long-term selection for carbon efficiency, redox buffering or rapid generation, rather than a single optimal strategy. We've then plotted all these parameters against each other to find out how they correlate. In the center we have length and weight, so that's pretty obvious, but basically any of these circles that are larger and redder mean a stronger correlation, and smaller and bluer, a larger or weaker correlation. So this is ambient conditions for our brook trout. The only thing I want to point out is we've also looked at how these parameters change as a function of being exposed to elevated temperature and hypoxia. And these were fish that were used in Connor's work that he had presented yesterday. And I'm not going to go through in detail here, but I just want to point out to see the differences. We're seeing distinct differences in the biochemical relationships as fish are exposed to elevated temperature. And then further on in the blue at the bottom, this is elevated temperature plus acute hypoxia. It's not simply an exacerbation of the changes that are under ambient temperature, but these relationships change when we add hypoxia to that mix. So there's a lot of things that we don't know yet, and a lot of things that we're excited to learn more about. But some takeaways are that ultimately we want to consider thiamine deficiency in context to be able to be used biochemically. Might have some more value. And again, this isn't really relevant in thiamine so much in eggs because this transplase isn't really developed yet. But then also thinking about how these pathways vary with each other. Do these associations between biochemical and morphometric parameters vary depending on exposure? And then acute hypoxia develops a separate health profile than elevated temperature alone. So we're really excited to expand this to other species, and excited to find collaborators interested in this line of work to look at other exposure conditions. So overall, I can't thank Eileen Randall enough, who's been instrumental in a lot of this work, Connor, Cliff, folks at the AFS, and a couple of undergraduate students in my lab, Panchika Mangal and Dennis Connor. Thank you very much for your attention, and I can be contacted via email address below.